that and hurling are the two games that they play. So yes, but it's interesting why suddenly they dropped the rugby and they kept the football. And football in this country, you don't use the foot for much.
No, just once in a while they kick a field goal.
So, should we? Okay. Yeah, let's do it.
Yeah, I just checked mine. It is on.
Don't forget, I can record everything you guys are talking about. There's open mics.
So you want them on or off?
Well, you might forget to turn them on. If you're not talking, I would just turn them on now. Or talking, wait till later. It's just one simple switch.
When we're up there, we'll be able to use that mic.
What's that?
When we're up there, we can use that mic.
If you're standing behind.
We only need these mics for the panel. But do we need to?
When you're up here, you can leave them on. Also, but you can also turn them off. I would just leave them on the whole time, actually. Doesn't really hurt.
Good evening. I don't know, maybe it's better if I talk. No? Does it sound like? Now. All right. Good evening, and welcome to the Universe Revealed speaker series. And so this is a series of lectures that's being given as a partnership between the St. Joseph County Public Library, Indiana University South Bend College of Liberal Arts and Sciences, and also the University of Notre Dame, the College of Science. And tonight, we're going to be having a talk about the Nobel Prizes 2022. And just to set this up, Alfred Nobel was born in 1833. He passed away in 1895. And he developed dynamite as well as stronger explosive devices. And he, in the patent for dynamite and for bombs and so forth, he made a large fortune. Before his death, he created a trust that became for the Nobel Prizes. And so in 1901, so that was 5 years after his death, prizes were awarded in physics, chemistry, medicine, literature, and peace. And then in 1969, a prize for economics was added. His intent was to award the Nobel Prize to those who have conferred the greatest benefit to mankind. Between 1901 and 2021, so that's not including this year's winners, 609 Nobel Prizes have been awarded to 975 laureates. The prize cannot be shared among more than 3 people or 1 organization. And the laureates receive a gold medal, a diploma, and 10 million krona, which is, in at least today's dollars, was roughly 800,000 US dollars. So tonight, we have 3 panelists. Our first one will be Professor Casey Clear, who teaches organic chemistry at Indiana University South Bend. Speaking for physics will be Antonio Delgado, who's a professor of physics at the University of Notre Dame. And then Professor Michael Frender, who's also at the University of Notre Dame. He directs the Genomic Core program and is also in the Department of Biology. So with no further ado, I'm going to hand it over to Casey Clear to talk about the Nobel Prize in Chemistry.
All right. Good evening. It's really exciting to be here and to talk about these great awards. So this year's award was awarded to Carolyn Bertozzi, Morton Meldal, and Barry Sharpless. And these 3, their contribution was for the development of click chemistry and bioorthogonal chemistry. And so these 2, well, I guess what I want to do today is give you a little bit more information on what click chemistry is, what bioorthogonal chemistry is, and why these are important in the first place. And so one thing you need to understand is that making molecules in the chemical sense, where we're we have these building blocks that are brought together to make new bonds, and building up of sort of collections of atoms is expensive. It's difficult. It takes a lot of technical expertise. You got to have specialized conditions. And Barry Sharpless, one of the Nobel laureates, was making the case for this idea of click chemistry, which is sort of an easier, more efficient set of reactions. And to justify this, he used just one example of this antibiotic called meropenem. And this is an effective antibiotic. It was discovered, small quantities were made, but it turned out that it was not, that it was a lot of work to actually synthesize to be used as a drug that everyone could use and buy in sufficient quantities. And so it took nearly 6 years to develop this. Right? So there's a lot of investment that goes into getting all these reactions to work. And chemists tend to look at structures like this, where we have these skeletal structures, and particularly organic chemists, or we're looking at these places where there are carbon atoms that are linking these together. But what you also should know is that molecules have three-dimensional shape, right? So this is sort of a representation of this antibiotics, at least one of the shapes that it takes on. So Sharpless laid out click chemistry as an ideal for chemical reactions. These are reactions that will work with simple reaction conditions and high yields. That means that basically if I mix this compound together and this compound and they form a bond, all of this molecule, all of this and this molecule will come together and be joined. There won't be side reactions and other material that has to be removed. There will be a high level of efficiency. Actually, I see some of my students here, and they know that from their experiences in the lab that this does not happen. It is not a given for most reactions that you're going to see high yields. So if you have byproducts, you want to be able to remove those easily. We need fast reactions, and importantly, the products that are stable, so that once they're made, the reactions aren't going to go in the reverse direction. And so a reaction that Sharpless mentioned that almost met this criteria is a 1,3 dipolar cycloaddition. So cycloaddition means that we have a ring, or we're forming a ring. That's where the cyclo comes in. Addition means we're adding two molecules together to make a ring, right? And this particular cycloaddition is between an alkyne and an azide. So an azide is when we have three nitrogens attached in a row. You've probably experienced or seen azide before if you have ever seen an airbag. So airbags have solid sodium azide that once detonated will rapidly expand, releasing nitrogen gas, right? And so that's why the airbag fills. I think I read that that maybe isn't as commonly used in most modern airbags, but very common in the 20th century. So these azides are basically primed to react, and they'll react with alkynes. Alkynes are molecules that have a triple bond between two carbons. If you ever heard of acetylene is the simplest alkyne that has that sort of structure used in welding torches. Now there is a problem though. This does not meet all of these criteria for a couple reasons. One is that this Greek delta symbol here indicates heat. That means we have to heat up this reaction to make these two products. And the other problem is there are two products. These are called isomers of each other, so they differ in their structure. So whatever this group is that was attached to the alkyne and the group attached to the azide, those are going to be taking on two different arrangements. And if you want a clean reaction, that's not an ideal situation. The next discovery that was shared between Meldahl and sharpless is very important, and that is a catalyzed reaction that uses a copper ion. So copper is able to speed up this reaction to the point where it happens at room temperature. It can happen in water. It's not super sensitive to oxygen and other things. So you don't have to have ridiculous conditions to make this work. It tends to work very well. And that's why it's called a click reaction. Now, what's very interesting about this is Melville's discovery of this was an accidental discovery. It came from a failed experiment. Melville was actually trying to make a big library of drug molecules, because if you want to make new molecules and test them to see if they're effective as pharmaceuticals, you've just got to make a big library of molecules and test them. So they're in the process of doing that. He was trying to react an alkyne with a different type of functional group that is a group attached to the molecule, but that molecule also had an azide on it. And what the product of this reaction was this, what we call a triazole, that cycloaddition product. And so it was a failed experiment, but it revealed something very interesting, a very useful reaction. And so both of these reactions, or both of the laureates, Sharpless and Melville, discovered this actually around the same time, that copper can speed this reaction up. And so this reaction is fast, it works at room temperature, and then we can easily remove, typically, the copper catalyst at the end. Now, why is this important? This has the ability to bring molecules together is important. If you want to do a modular sort of synthesis, so just for an example, polymers, which are a series of small monomers, that's what we call the single units that have been joined together to make a long unit. Now, you may want to be able to easily add a second molecule to those. So if your polymer has sort of a handle of an azide or an alkyne, you can go ahead and add those at the end. It gives a lot of flexibility when you're doing synthesis. The high yield and easy conditions make it a good reaction for pharmaceutical discovery. And so there are numerous uses that this idea and then this specific reaction has played. So the last thing I want to mention is this idea of bioorthogonal chemistry. And so this was the contribution of Carolyn Bertozzi. And so this is from her paper. This is reactions of functional groups that are so selective for each other that they can be ligated or brought together in a richly functionalized biological milieu. Basically, in a biological system, we've got all kinds of stuff. We've got water, we have proteins, we have all kinds of different structures that could react. And so the bioorthogonal reaction is one that doesn't react under those conditions. It's a separate set of reactions. And so the reaction pair that she developed was this alkyne and azide pair, right? Azides are not normally formed by biological systems. Alkynes are not a common feature in biological systems. So if you are able to sort of introduce an azide into some structure, right now you can add this alkyne in there, and this high efficiency of this reaction will make it work. Now, of course, in this case, copper is toxic to cells. So if you want to go into cells, and this is one of Bertozzi's contributions here, is that instead of the typical azides, these azides that are trapped into the rings, and they're basically spring-loaded so that when they meet an azide, they're able to react. And so this is called rather than being copper catalyzed, these are called strain-promoted reactions. So the beauty of this process is that some of the biological machinery was used to make these, to introduce these sugar molecules that contain azide functional groups. And then when the alkyne was added in, it was able to label that group through this highly efficient click reaction. So in this example here from one of Bertozzi's publications, this shows a fluorescent dye that has now labeling any place where this glycan has been introduced. And this can be done with minimal disruption to the cell, so you're kind of able to stealthily watch what's happening in the cell by adding these labels. It's also this type of being able to do these reactions can be used to make new sorts of drug antibody conjugates or other things that can be used in anti-cancer treatments. And so there's work being done on that. So, and the way that this sort of the biochemistry of the cell was able to be interrogated here with these methods has really given rise to a great number of sort of byproducts, new areas of direction. And so that's part of why Bertozzi has been awarded, is a laureate on this award, because it has changed the way these our understanding of science, understanding of cellular processes works. So I hope I have, at the very least, you have an idea of now click chemistry, what that means, and a little bit of understanding of this bioorthogonal chemistry and some of these contributions. Thank you very much. And I believe we'll be taking questions at the end.
So, hello everybody, I'm Antonio Delgado. As you can hear from my accent, I'm from Wisconsin. So, I'm gonna give you a short introduction to the Nobel Prize in Physics that was given to Alain Aspect from France, John Clauser, American, and Anton Zielinger from Austria. But in order, and the prize is given 1/3 to each of them for the foundation or the experimental foundation of what is called quantum entanglement. So in order for me to be able to give you a picture of what is quantum entanglement, I have to go back in time and go to the end of the 19th century. At the end of the 19th century, physicists thought that they have already understood the world. They had a way of understanding how things move. This is what we call mechanics. And the foundation of mechanics comes from the Greeks, from Galileo throwing things from the Tower of Pisa, and from other more mathematical works, but in general we understood that things move because of forces. Planets were understood by Copernicus, Kepler, and Newton gravitational theory, and it was very easy to prove that the planet goes around the sun and not the sun around the planet. So, okay, that's understood. And then light was understood as an electromagnetic wave thanks to the experiments of Maxwell and Gauss and Franklin, including Franklin. And so they had more or less a robust understanding of nature. And yes, there were some things here and there that they weren't clear, but they thought that, okay, physics is done. We just have to do more careful measurements and we have understood how the world works. Well, that's not what happened. At the turn of the century, two theories emerged to explain that no, what you said is completely wrong. One is the theory of relativity, and that's the foundation of special relativity and then later general relativity. And that's based on the fact that an experiment trying to discover something called the ether, that I don't have time to talk about, but I can take questions. They didn't find the ether. What they found is that the speed of light is the same. The speed of light doesn't change. Well, that made Albert Einstein propose his theory of relativity, who changed somewhat, especially mechanics. But it wasn't such a big change. The biggest change was when, starting with Planck, but also Einstein, started analyzing light. And light isn't or wasn't what they thought it was. If I take light and I put it through a prism, well, we have done that, all of us, and then you get a wonderful rainbow, all the colors. But that is what happens if you use the light that now doesn't exist, because we are not allowed to have normal bulbs anymore. We have LEDs. But if I would take a light of a bulb made with sodium inside, I will get a very yellow normal light. If I put that into the prism, instead of getting a rainbow, I will get two very sharp lines, very sharp and very clear. And that happens with all elements in the periodic table. You get spectra. Light is not continuous. Light has spectra. The solution that Planck, Einstein, de Broglie, among others, was that, okay, light is not actually a wave like what happens in water. It's made out of smaller objects called photons. And these objects, they behave very weird, in a very weird way. Because sometimes they can be waves, and sometimes they can be particles. This is what is called the particle-wave duality. One of the consequences of this quantum mechanics that is very strange, and it has implications very profound. One is indeterminism, another one is two objects cannot be at the same time in the same place, and we can discuss that. But one of them is that I could create pairs of these two photons that they are together, and they are called entangled. And you use the word tangle because they form a pair. So I form this special pair, and then I send this photon very far away. And suddenly, if I measure one property of the photon, which is the polarization is anybody who has sunglasses knows what polarized mean. So light has two polarizations. If I measure the polarization of this photon, I can tell my friend Pat, which is there, that I can predict what is the polarization of the photon he has. Einstein didn't like that. Well, not just Einstein, lots of people. And Einstein called this spooky action of a distance, saying, okay, how on earth can it be that I can predict what Pat is going to measure just by measuring this? So in 1935, Einstein with Podolsky and Rosner wrote a paper called EPR, suggesting that there is a way that you can prove that this is right and wrong. And the theory they suggest to avoid this issue is something called hidden variables, is that this pair of photons, and we don't know enough about them, so there are some hidden property that I haven't measured. That's why I'm able to make predictions. I'm able to make predictions because I don't know enough about this system. Later in 1964, John Bell, a theorist, he analyzed this idea by Einstein, and he tried to work, how could this happen? What would it take for a system to be a hidden variable versus quantum mechanics? So he introduced two observers that they are called Alice and Bob, and since then everybody that works on Bell's inequalities call it Alice and Bob. So you can show that I'm Alice, Pat, you are Bob. You can show if there is a hidden variable And it's a very easy proof. You just need to know algebra because you're doing probabilities. And you can show that if there is a hidden information that neither Bob nor Alice know, but you can prove inequality saying that the probability of making a measurement is bounded. So it has to be bigger than something else. So John Bell said, okay, if you make an experiment and you violate this inequality, then quantum mechanics is right. If you don't violate this inequality, then these hidden variables are the right explanation. Of course, you know the answer. Quantum mechanics is absolutely right. And first Closer, in 1969 to 1972, he envisioned some kind of experiments, which they are very easy experiments. You just send in 3 lights, 3 lasers. And he was just checking that these entangled pairs, by knowing or by measuring one of the polarization, he could predict the other 2, violating completely Bell's inequalities. So quantum mechanics is strange. Yes, is entangled. Yes, there are no hidden variables. That's good, because if the solution was that there were hidden variables, they are hidden. So you have to measure them. So that was 1969, 1972. Then Alan Aspect, in 1981, 92, he did other experiments complementing what Closer had done in 1972. And finally, Zeilinger closed all the loopholes in 2012, where he was able to show disentanglement over 143 kilometers. For those of you who don't understand kilometers, this is 90 miles. So you can send the prediction over very macroscopic distances. You may say, okay, who cares? Well, I care because I'm a scientist and I don't know if what I do is useful or not, is useful for knowledge. But these entangled pairs, these things that you can create a system that when I separate them, they still know about each other, is used in encryptation. Is used to have what they are called quantum passwords. And these are used in Switzerland for people that have accounts in banks that none of us can access. But in this way, the bank can give you a key, which is a quantum key, and until I measure it, then I don't know about the other key. But once I measure it, I know exactly what is the other key. So I can have very effective encryptation. It's unbreakable because I am the one who has one of the keys. Of course, it's unbreakable if you, like me, are a theorist. The problem with these entangled pairs is that they work perfectly if there is a vacuum. At the moment that there is something else going around, this is what is called, in quantum mechanics, decoherence. And then the entangled pair is broken. So you have to make sure that you keep the coherence, that is, that this entangled pair is always entangled. Not completely unrelated to this is a notion of quantum computing that you may have heard in the news. You create also this set of entangled objects, they are called qubits instead of normal bits. And as long as they behave like in quantum mechanics, they can be at the same time in the 0 and the 1 position, which is the usual classical computer. The biggest problem is also decoherence. Heat, that the previous speaker was talking, is horrible for decoherence because it breaks that. So this is more or less the reason why this Nobel Prize was given, is a fundamental property of quantum mechanics. But also I have to remind to you that one of these people did the experiments in 1972. So it takes a long time. And one thing the host forgot to say is that you have to be alive to get the Nobel Prize. Yes, no. And let me finish before moving it to biology, is that there have been cases, at least that's the legend, that the Academy has waited for one of the people to die to go to 3. Because there were 4 people and they couldn't decide, and they say, okay, time is the thing that is going to solve the issue. Okay, as the previous speaker said, we will take questions after the 3rd Nobel Prize. Thank you very much.
I always like these things because I learn a tremendous amount from both of those speakers. So thank you so much for inviting me to come today. Yeah, here we go. So it's a real joy to be able to talk to you about this particular Nobel Prize, because in medicine and physiology, typically the prize that's given out has some fundamental biological property, something that's directly related to human health. An evolutionary biologist is sort of an oddball in this collection. And since I'm an evolutionary biologist, I'm particularly fascinated by this one. And so Svante Pääbo, this smiling, good-natured looking fellow, wouldn't you give a Nobel Prize to him? I would. Apparently the committee thought so too. He is a really good evolutionary biologist who's brought his skill set to bear on some very large problems that influence all of us. And so I'll try to illustrate some of the impacts that he's had as I go here. So in order to understand sort of what he did and why he got this award, think about this statement for a second. So something like 1 to 4% of the DNA in many of our genomes is actually derived from Neanderthals, a closely related group of humans. Many of us have actually got this result. People can now get personal genomes and do 23andMe, and so you might read the report and presto, there you are, you're part Neanderthal. The thing to think about is, how would we know such a thing? Like that's a pretty profound statement. What would you need to be able to actually say something like that with any confidence at all? Just think about that for a second. What sort of technology would you need, or what information would you need? 20 years ago, you wouldn't be able to do this at all because some of the essential pieces weren't there. One of those essential pieces is you need a really well-resolved human genome. And this is something that was first published in 2001. In my estimation, perhaps one of the most profound scientific discoveries of this whole century happened in the very first year. That's an interesting thing to think about. In the 20 years since then, we've really come to understand what's in the genome and what the content is and what the genes are. And so this is the framework that you need to be able to answer a question like on that last slide. That's one thing. The other thing you need is the technology to actually get DNA from ancient specimens, really old specimens. And it turns out that this is not at all trivial. And this is the area that Svante Pääbo really excelled and pushed the science forward. And so to tell that story, you have to go back to 1985 when he first started, not 1972, but still they wait a long time to give people these awards. In 1985, he published a paper where he looked at DNA that was extracted from Egyptian mummies that were on the order of 2,400 years old. So think about how challenging this is. Egyptian mummies are subject to all kinds of chemical fixatives. There's all kinds of organics on them. They're sitting around for 2,400 years. The DNA that you get out of that is going to be in minute quantities, and it's going to be highly degraded and fragmented. So the best thing you could ever hope for is to get a bunch of little tiny pieces of intact DNA. And that's exactly what he did in this paper. Right away, 2 problems became apparent. One big one is contamination from modern humans. So in contrast to the Egyptian DNA, the DNA of everybody handling the specimens and working around them is fully intact, highly abundant, there's lots of it. It's easy to amplify with polymerase chain reactions, it's easy to analyze. It takes over anytime you try to do this. So one of the first things they had to do was figure out how to deal with this contamination issue. And the upshot of that was, over the next 10 years, is people started working in labs that were very much like the labs you'd see for highly infectious pathogens, where everybody was in white suits, there was air supplies, air locks, ultraviolet lights coming on at night to get rid of all the DNA. And so what happened after this sort of experiment and seeing these weaknesses was this whole area of ancient DNA, which we would now call paleogenomics. That field really took off and matured over the next 10 years or so and became what it is today, which is a really rigorous field. So think about those problems for a 2,400-year-old sample, and then think about this question of Neanderthals to get back to them. To analyze a Neanderthal sequence and to think about its relationship to modern humans, you have to be able to look at samples that are 40, 50,000 years old and have been in the ground for a long time. And any of the available samples have been in museums for decades with people handling them without gloves and hopelessly contaminating them. So Svante's group was really confronted with a major challenge in trying to do this. But the questions are really obvious. Like, what are Neanderthals? Are they just a variant of modern humans? Are they really just us but look slightly different? There's lots of variation in human populations. Or are they something completely different that had no genetic interaction with modern humans? Or was there interbreeding and some genetic interaction? These are really important questions if we're going to understand the origin of modern humans and what our history really looks like. In the late 90s, I think 1997 was the first publication, the first sort of fragments of DNA from Neanderthals were coming out of Svante's group, and we were able to ask some of these questions. And right away it was obvious that Neanderthals were quite different, well outside the variation that we see in modern humans. So they're different. The small pieces of DNA that they could analyze weren't enough to ask the question of, was there interbreeding, was there real genetic interactions? It wasn't until 2010 when they were able to sequence the complete genome and take all those little tiny pieces and put them together into a complete genome sequence that then matched the human genome. The other advance that had to occur, aside from the technological side of it, was the computational side of it to really take those tiny little fragments of DNA and assemble them into a whole genome. So there's this whole computer science side of it too, and Svante was in charge of that. Here's a couple of pieces of the actual bones that were some of the first ones that they got DNA from and led to the complete genome sequence that was published in 2010. The punchline is that it's clear that there was interactions between humans and Neanderthals, and we can see the traces of that in our own genomes now. And this became apparent right away when we looked at this genome. So now the machine is there, right, and the ball is rolling. And so within 2 more years, and even what I think of as a more remarkable finding came about, that came from this tiny little bone. This is a small piece of a finger bone from a young female that was found in a cave in Central Asia, the Denisovan cave. And Svante's group was able to sequence a complete genome from that little tiny piece of bone. So that gives you some sense of how far the technology has really advanced. And the remarkable thing about that is that this was a completely different lineage, not Neanderthals, not modern humans, as different from either of those as Neanderthals and humans are different. And so now we get this remarkable picture that in our recent past, we weren't the only ones. There were at least 2 other groups of humans that were quite distinct from us, that were occupying all of Asia and Europe, the same place that we were, and so we were interacting and co-occurring with them. And we were interbreeding to some extent with both of them. This plot right here shows the distribution of Denisovan alleles in human populations, and you can see that in New Guinea, for example, there's a quite high frequency of those alleles. So if they have 23andMe done, it wouldn't say they have Neanderthal, it would say you have 1 to 4% Denisovan alleles. Quite different. So the upshot of all this work is that we now have this much richer sense of what our recent history was, where something like 400 to 1 million years ago, there's a split among lineages. This group goes to Neanderthals and Denisovans. This group ends up coming out of Africa and being what we think of as us, modern humans. But there's genetic interactions between all of these lineages. And that legacy is still in us. We still see that today. And so it's a much different picture than we would have had prior to this DNA work when we just had a few fossil bones to look at. Svante gave us that view of our rich, rich history. So the question is, well, why do we care about this? What's important about it? We learn a lot from these genomes, aside from just this historical perspective of what makes us and how did we get here. There are lots of things in Neanderthal genomes and Denisovans that are useful to us. There are lots of things that are detrimental to us. There are genes that affect our immunity system. There are genes associated with lots of diseases like osteoporosis or blood diseases. A recent study came out just within the last couple of weeks that showed that there are unique mutations in humans that cause increased growth of neurons in developing brains that Neanderthals didn't have. And so that suggests that if we know what's in Neanderthals, we can start to tease out what makes us different. How did we get to be the special individuals that we are with skills and capabilities that clearly no one else had? And so it allows us to think about how did we get to this stage where we have things like culture, where we actually have this explosion of intellectualism that no other animal on the planet has ever had anything close to that. So we owe all that to Svante. Many, many other people contributed to this. But one of the true, I think, signatures of his impact is if you look around and you ask, who are the leaders in paleogenomics? Who are the big shot professors all around the world? Almost without exception, they came through his group. And he sort of trained this whole cohort of evolutionary anthropologists that are now disseminating this and advancing it. And the techniques of ancient DNA go widely beyond looking at humans. We are now looking at the DNA of extinct animals to try to figure out what they were like and could we perhaps resurrect them. It's just a routine thing now to look at old DNA. But when Svante came on the scene, it was a mission impossible. No one thought you could even do this. So I'll stop there, and I guess we'll all be happy to take some questions.
We should wait until, are you going to give her the microphone?
I think what I'll do is I'll repeat your question.
Who is the panel that decides the winners in the categories? How are those people chosen, or who does it? Who does the deciding?
Do you know? I only have a vague understanding of that.
It's not public. It's not public. There is something called the Nobel Committee, but it's a combination of what I have here is previous winners, previous laureates, and other people. You can send names to the Academy, and then the Academy, I think it acts more or less like a black box.
Mm-hmm. But there are experts that are sort of senior scientists, because I know a few people who have been invited occasionally to participate. They're not regular members, but they might be called for some particular area.
Yeah, and I believe the science awards, I think if you're familiar with the Nobel Peace Prize, you're
probably familiar with Oslo,
Yes.
Norway, but whereas like the science prizes, I believe those are all based in Stockholm.
Yes, all of
Sweden,
them.
And I think it's the Swedish Academy.
Well, but there is one reason why the peace is in Oslo. When Nobel was alive, Norway and Sweden were the same country. So that's why it's in Oslo. Yes, there are experts that they get emails and also they have to sign a confidentiality agreement. But I have to tell you that since I remember, all the Nobel Prize of Physics were well deserved. I don't know if my colleagues have the same opinion, biology or chemistry, but
I think at least all the recent ones I can think of were highly deserved. In fact, as you alluded to, sort of past deserved, right?
yes.
It seemed like it took a little too long to get that recognition that they deserve.
The question, what about the recipients? How do they get to be examined? Are they nominated? And is there more than one group?
Well, it depends. I don't think there is a pattern. You can be nominated, the Academy may have heard about you. The only condition is that it's a maximum of 3 people, but the Academy can decide how to share. For example, in the physics one is a third this year, but I remember in other occasions where there were 3 people, but one got a half, and the other one they got to share a half. And the Academy gives a reason. There is a reason if you go to the webpage why they have given, but you cannot appeal.
So in the chemistry one, those people didn't necessarily, those scientists weren't working in tandem with each other, were they?
Not directly. Yeah, the Sharpless is actually in the U.S. and Meldal is in Denmark, and those 2 discoveries is very interesting. They reported them the same year. I think it was 2001. Those discoveries of that catalyzed reaction, and Bertozzi is a sort of a biochemist and chemical biologist, and so there was not a collaboration. They shared the award because they all contributed to this important discovery of click chemistry, which then extended into the bioorthogonal chemistry.
First talk, the click which you showed a slide, and there was the copper catalyst. When I was looking at the slide, it seemed, was the big benefit there was that there was one product that was better, more of a product than it used to be without the catalyst? In other words, there were 2. Yeah, there were 2.
Yeah.
Did it go towards one of them rather than the other?
Yes, you were paying attention. Yeah. So the 1,4, I believe the 1,4-triazole product was the one that was favored. That reaction was catalyzed, whereas the other reaction was not catalyzed. So that was the speed. The fact the reaction was sped up was very important, and the fact that now it only produced one isomer or one form of that molecule was very important.
How does one go about putting together random fragments of DNA to create a whole genome of an extinct species?
You first get the biggest computer you can find and the smartest person you can find, and you put them in front of it. There are lots of different techniques to do it. The easiest way to think about it is like a jigsaw puzzle, where you take a piece and then you look for something that overlaps it just a little bit. And this is called sort of a consensus overlap method. And you just keep building bigger pieces. In the early genome, like when the human genome was made, they went to great lengths to make different size pieces. So little teeny pieces for close stuff. And then they would make pieces where they knew the ends but didn't know what was in the middle to try to bridge pieces together. And so it's almost a brute force kind of a thing. If you have enough of the little pieces and there's enough overlap, and you have lots of computer time because you got to shuffle all these pieces and check them all together, you gradually can build up a genome. It's a little easier now that we have a reference genome that they already did that to, because now you can take those pieces, you can ask, well, where do they live? Where's their best match? And so it's much quicker to match them up to the reference genome and then put them together and sort of pull away this complete template. But it takes lots of computer power and lots of data.
I have a question for Neanderthal as well. So theoretically, the direct offspring of Neanderthal and our ancestor should be 50-50, right? 50% Neanderthal DNA, 50% our ancestor. So how long does it take, after 50,000 years, now we have 4%. That's a long time for us to still have 4% of a Neanderthal gene. So I'm just wondering what is going on, how many generations, and then what else is playing a role in us having 4% left?
I think that's an excellent question, because you would think after that much time that it would have just sort of been diluted out. There's no more Neanderthals to keep putting more back in. And so what that suggests is that it's not just neutral, but that there's actually selection that's promoting and keeping some of it around. And I think that's one of the interesting things that people are trying to figure out is, are there advantages to having some of those pieces that would make them better than some of the other gene variants in other humans? And a good example of that is during the COVID epidemic. There was a couple papers that came out, Svante was the lead author on them, that were looking at how genes that are associated with resistance to COVID or the milder symptoms when you're exposed to COVID, did any of them have an origin that was Neanderthal or not? And more than you would expect just by chance did. And so that suggests that there was some advantage in that particular context for some of these variants. You could also find variants that had a negative relationship and would be sort of pushed out of the populations. But I think that's a really insightful question, is to ask, why do we still have that many? And it suggests that there is more going on to those genetic elements. They're not just pieces of junk DNA floating around.
Just a general question about competition for these prizes. Are these people even aware that they're in the running for it, and how many are in the running for these prizes each year? Can they be selected year after year?
I can only speak for myself, but I always wait for the call, and I've never received it. And so I'm not sure what the process is, but...
Some people know that it may happen, but there is very little competition because the Nobel Prize is awarded for things that were done 20 or 30 years ago. So there is no actual competition now. And yes, people have claimed that, oh, this person was about to, but maybe next year. But this is all gossip. There is no webpage you can go and check if your name is there or not.
But there is a fair amount of speculation in the scientific community, like who would be a likely candidate next year, like who hasn't got one and is deserving. And I think sometimes you can figure that out, and sometimes it seems like it's very hard to figure.
Any other questions? Well, thank you very much. I think let's thank all our panelists. We'll see if this rings. So we have a couple of events coming up. So next Thursday at the University of Notre Dame, there's an inaugural science prize that's actually being given to the 2018 Nobel laureate who won in physics. So Professor Strickland will be speaking next Thursday at 3:30 in the Jordan Hall of Science on the Notre Dame campus. And she's helped change really the field and applications of laser physics. So that's a nice piggybacking on tonight's talk. And then also the week after, so on Tuesday, November 8th, we'll have another Universe Revealed speaker. So this is Ilan Levine. He's at the Indiana University South Bend in the Department of Physics and Astronomy. And he's going to be talking about dark matter particles. And so these are undergraduates and high school students who've been working with him in the search, apparently with big heavy tools. So please join us. So next Thursday, Notre Dame campus, Nobel laureate 2018 physics, Jordan Hall of Science, and then Tuesday, November 8th, back here. And thank you so much.